Hello, good morning everyone. Welcome to my channel. Today's topic is photosystem. You know that chloroplast contains grana. Individual sacs of granas are called thylakoid. Thylakoid is a membrane. Membrane is made up of phospholipid bilayer. In this thylakoid membrane, there are special proteins called photosystems are located. So location of photosystem in plants and algae is thylakoid membrane. Whereas if you consider photosynthetic bacteria, in them you will find photosystem in their cell membrane. What is the role of cell membrane, what is the role of photosystem, and how the structure of photosystem is? That's what we are going to understand in this particular topic. Photosystems are of two types, photosystem 2 and photosystem 1, which is named based on discoveries. If you take up one photosystem, if you enlarge it, this is how the photosystem looks like. It is having sort of three chambers in them. In two peripheral chambers, you will find many pigments associated. About 200 to 400 pigments are found. Those pigments are called accessory pigments. Which and all pigments are there? There can be chlorophyll B, xanthophyll, carotenoid pigment. So what is their role? Their role is to absorb the light or harvest the light. So those pigments are called light harvesting pigments. Together, light harvesting pigments along with proteins form light harvesting complexes. Therefore, in photosystem, there are two light harvesting complexes made up of light harvesting pigment. So, I have given you the classification. Photosystem is composed of light harvesting complex, in short LHC, which is composed of light harvesting pigments associated with proteins. Light harvesting pigments are also called accessory pigment. They are also called antennae. Why are they called antennae? Because sunlight will be consisting of ultraviolet light, infrared, visible light, all those lights. These pigments are like antennae. They collect only specific wavelength of light, that is from 400 to 700 nanometer of visible spectrum, which is required for photosynthesis. Hence they are also called antennae. Then coming to the middle part of the photosystem, it is made up of a chlorophyll A pigment. This chlorophyll A pigment forms the reaction center. Apart from pigment, there is an electron acceptor also. What is the difference between chlorophyll A pigment present at the center to that of pigments present in the light harvesting complex? Pigments present in the light harvesting complex can collect the light energy, and they cannot convert light energy to chemical energy. Let us understand what exactly is happening. When pigments first absorb photons of light, their electrons jumps from ground state to the excited state. From the excited state, electron comes back again to the ground state. It releases energy. That energy is transferred to the adjacent pigment. So there will be a continuous energy transfer in accessory pigments of light harvesting complex. Finally, all the generated energy will be collected by reaction center, which is nothing but a chlorophyll A molecule. So what we have understood is accessory pigments can only collect the light and transfer the energy. They cannot form any products out of it. Whereas chlorophyll A, which forms the reaction center, through the it will collect the energy. Now its electrons are excited, which will travel, which is collected by an electron acceptor. Then it will travel through ETS located in the thylakoid membrane. It ends up preparing NADPH and ATP, which we can regard as chemical energy. So that is the difference. Accessory pigments can only collect the light energy, can transfer the energy, whereas reaction center can convert the light energy to a chemical energy like NADPH and ATP. That is the major difference. So this is about the structure of photosystem. In bacteria, if you consider, light harvesting complex is composed of only 20 to 40 pigments. I hope you have understood the concept of photosystem. If you have understood, please click the like button, like, share and comment. Thank you.